ladies and gentlemen, so just a friendly reminder, your final, if you're in WI, your final reviews were due Tuesday. If you haven't handed those in, do them now. And we'll get the peer review assignments out to you by the end of the day, so you can turn those in. If you are not caught up on any of your writing assignments, please turn them in. You can turn them in up until the end of the class, and you'll still get credit for having done them, even if you don't get full credit in terms of points. You can still get your WI accreditation, which is worthwhile, I think. And just another reminder that your final projects are due April 20th, that Tuesday of class. If you have something digital, you can email it to one of your instructors. Otherwise, we'll have a big cart with physical things that you can pile onto it that we can grade and get back to you, hopefully by the time you take your exam. So, yes?
Isn't that on a Thursday?
I don't remember. It's on the,
I'm pretty sure in the syllabus it says the 21st, which is Thursday.
21st. Is that Tuesday or Thursday?
Next Thursday.
Thursday, the 21st. Okay, so I am so pleased to be able to invite for the first time to this class Dr. Matt Lacchei, who got his BS at Cornell, upstate New York, and then came here to study connectivity and population dynamics of invertebrates, focusing on lobsters. That's not the only thing he studies. He works on anything that gets wet in the ocean, invertebrates, vertebrates, genetics, you name it. Dr. Lacchei has done it. His main focus is connectivity, distribution, and diversity of marine organisms using a whole bevy of cutting-edge genetic techniques. He's moved on now. He's a postdoctoral research in oceanography department, and he's going to tell us all about what he's up to. Take it away.
Okay. Thanks, Anthony. Thank you guys. He works in the sea but has trouble with any electronics, just like all other professors. Today I'm going to talk to you a little bit about some of my PhD work, population connectivity of spiny lobsters specifically. I know you guys have been in the class. The semester's what, 2/3 of the way over or so? So I don't know. I looked through the schedule a little bit. You might have an idea of population connectivity. Does anyone want to start off on a guess what connectivity is, or what does connectivity mean to you?
Connection.
Connection of what? Your soul with another's? Could, right? What about population connectivity? You don't do that yet?
Like how one population.
Sure, in the back. Sorry.
Connections between different populations.
Okay, a connection between different populations. And what kind of connections could those be, do you think? Yep.
Gene flow?
Gene flow, sure. So that's what we're going to look at. I'm going to start with genetics, but it can be movement, right? It could just be individuals moving. They could eat in one area and crap in another area, and that's connectivity as well, right? So if we just start off with the basis of connectivity, it's just the quality or condition of being connected, just like you said, right? So this is straight out of the online dictionary, right? But population connectivity is this exchange of individuals among, generally we're talking about geographically separated subpopulations. And the reason we look at this, does anyone have an idea of why you might be interested in this? I mentioned a couple of things, right? You eat in one place, you shit in another. That affects the ecosystems in two different places, right? Gene flow, so you reproduce here, your child moves to the mainland, now there's gene flow to the mainland or to another country, right? So in ecology, population connectivity is an essential piece of knowledge to understand things like recruitment, so where are larvae settling out, or where are those juveniles moving to, predation, so reproduction, speciation, so all of these phenomenon operate on specific scales, right? So if an organism moves off of Oahu and moves to Maui and feeds there, that's predation that's moving from Oahu to Maui, correct? So this also drives more broad processes, both in temporal scales and geographic scales. So essentially looking at speciation, biodiversity, so who can you mate with, where do you live, that drives the number of species in a certain location, is driven by population connectivity, right? And it also, in terms of management, so a lot of the questions I've always approached in terms of fisheries management Population connectivity is important for looking at stock structure, or if you want to do marine reserve design, and looking at just general energy flow through ecosystems. So from the most basic level, though, we want to know, how did species get here, right? So for speaking about Hawaii specifically, and then do they survive and establish new populations here and or new species, right? So Tuesday, Marguerite talked to you, and I'm sure she went through some terrestrial species radiations based on her general title talk, I'm guessing anyway. So I'll brush up on that, I'll talk about that very briefly. But for those of you who didn't grow up here, or those that did, and had to ever travel outside of Hawaii, you might have noticed that it's really far from anywhere else. And so this is a Hawaii central map showing distances of Hawaii to, I think LA is the closest, what's that, 3,750 miles. Quito, 6,700 miles, down to Santiago, Chile, 7,000 miles, right? So for any organism, besides maybe humans, and even for flights, right? What's the shortest flight from Hawaii that you can get to some other state or some other country? And it's like 5 hours flying to California, probably. So as I mentioned, when we look at terrestrial species, this has big impacts on what happens to terrestrial species when they get here, right? So rarely do they arrive here, so very few species from the terrestrial realm have arrived here. And then when they do arrive here, right, even there's multiple species from the same genus that have radiated even within an island, let alone on multiple islands as those islands have come out of the water, through geologic time, right? And I'm guessing you guys have touched on this a little bit. So here's a bunch of species that are endemic to Hawaii, so just found here, but once the first species arrived, radiated into multiple different species, occupying a variety of habitats. And so in general, this is, I think, I got this number still from the 80s, from I don't know if you're using the Natural History of Hawaiian Islands book, but approximately 90% of the terrestrial species here are endemics, and that's until we started introducing lots of other species. And generally, within one single genus, the average number of species in a genus is 10 for a terrestrial species, right? So in the marine realm, those same barriers to dispersal, right, between islands, those channels between islands that prevent organisms from moving between islands and foster that radiation into new habitats, is the medium in which individuals move, right? So we have much lower levels of endemism in reef fish species in Hawaii than we do in the terrestrial realm. So 30% approximately, and this has gone down a little bit, I think, since some new work has been done, but of the 440 inshore species are endemic. So now we're looking at comparing 90% terrestrial, right, with 30% marine. And the average number of species per genus, we're looking at about 2 to 3 rather than 10. For the reef invertebrates, it's still in the same realm, so 32% endemic, and a few less. The average drops a little bit for invertebrates for how many per genus are here. And so this is because for marine fishes, so like when we think about terrestrial species, you're looking at adults that are moving, right? So you got two of you decide to reproduce, you have a child, that child isn't dispersing long distances without you. And that's how it generally works for terrestrial species, right? So it's the adults that are moving between locations. In the marine realm, other than the charismatic megafauna that you think of, sharks, turtles, whales, everything else, pretty much, the adults have fairly small home ranges, right? So they don't move that far as adults. For those of you who've gone snorkeling or go on these snorkel tours or whatever, they bring you to Turtle Town, and there's probably 100 turtle towns in Hawaii, right? And there's always turtles there because they don't move very far. Even those turtles, they come back for food sources, and especially things like reef fish or lobsters, people have their specific fishing holes or that they always go back to because they know those organisms, that habitat is preferred habitat, and the organisms don't go that far. And so what does go far is the pelagic propagules, so the juveniles that are released into the open ocean. But obviously these are a lot smaller, and they're affected by those ocean current forces, and that sort of drives where they can go. And these propagules are what potentially link distant locations. And so despite Hawaii being that isolated, it's one of the most isolated archipelagos in the world. But there are currents, so this is a graph of ocean surface currents, and Hawaii is like right in here between the SEC and the North Pacific Current. The currents travel approximately 20 to 25 kilometers a day, and this is specifically this south equatorial current. So larvae would have to get trapped a little bit north of there to be in here, but if they were traveling from the East Pacific, they could come across the south equatorial current, 20-25 kilometers a day. And so generally marine species larvae range anywhere from maybe a couple hours to, well, some up to 4 years. So the longest larval duration is about 4 years. But most of them are within the couple weeks to a few months range. And so over a 3 to 6 month larval period, these individuals can travel 2,000 to 4,000 kilometers. And so the group of species I'm going to talk to you about today are the spiny lobsters. And in general, this is a graph of that larval duration, so how long larvae are in the water column. And so you see this is in days, so 0 to 600. And the lobsters and some other crustaceans are located down here, sort of at the longer end of the spectrum. And this is a phylosoma, which is what the actual spiny lobster looks like. And the slipper lobsters, they're crazy. Guys on long lines pull these up like long line fishing boats, and they can apparently get as big as this. I've never seen one in the wild. They're fast swimmers. They're predators even as juveniles. So they're pretty interesting larval stage, and they have some of the longest dispersal times in the sea. And so through this 9 to 12 month larval stage in the open ocean, so this is for the two species I'm going to specifically talk to you about today, they go through multiple molts. So they actually metamorphose every couple weeks to months into a new larval stage, and then the final stage they settle out, and they look like a lobster, juvenile lobster, except that they're clear and they're really small, and they tend to settle out in shallow water habitats. And I have a blank slide. Oh, here we go. And so that has allowed a number of lobster species to actually arrive in Hawaii, so to cross those broad distances and make it to the Hawaiian archipelago. And so this is just a pictorial showing you some of the diversity that we have here. And so we have the two spiny species. There's a number of slipper lobster species. These are the closest related. So this is Anoplometopidae. It's more like a crayfish, but they're actually the closest related lobster to what you would traditionally think of as a lobster. So most of you, when I say lobster, you think of two big claws, boiling in your pot, red from the Northeast. But those type of lobsters actually only live in the Northeast U.S. and the Northwest Europe. So there's two species in that genus. And the majority of lobsters around the world are actually spiny lobsters, and most of them are located in tropical habitats. And then also those slipper lobsters that you saw as we were going through. And so the two species that I focused on, or two of the species I focused on, the two in Hawaii that I focused on for my dissertation work, was Panularis marginatus and Panularis penicillatus. And these two offered an interesting contrast because Panularis penicillatus is the most widely distributed Panularis species in the world. So the species range goes from the Red Sea all the way to the East Pacific. So they're located in the Galapagos Islands. And few species span that range. So there's the majority of tropical marine species, I'd say, can be found sort of in between like the Marquesas, or not really the Marquesas, but like the Tuamotus or some parts of French Polynesia, west, even west into the Indonesia or the Western Indian Ocean. But the species that make it into the Red Sea and the East Pacific, I think you're getting down to below like a handful or a couple handfuls of species. So very few numbers span that range. And so you would think that population connectivity, right, that we're going to talk about would span that whole range, right, so that there would be connected populations. Obviously the larvae can make it there, and obviously the species is there, but are those populations connected? In contrast, the Panularis marginatus is an endemic species to Hawaii. Do you guys learn what endemic is? Yes, nods. Does anybody want to say it out loud?
But no.
No. Surprise. So an endemic species is a species that's only found here, right? So it's nowhere else in the world. So you have a lot of vocab, you know, native species is here, it started here and is found other places, but endemic is actually only found in Hawaii. So back to that slide of what's the percentage of endemic marine species, do you remember? 30%, right, so in marine inverts, 32%. So this is one of those species. The other differences between these two are the depth range. So Marginatus here can range from 5 to 300 foot depth, whereas Panularis penicillatus is really shallow. So 5 to 60 feet depth range, but in general, you're not going to find them not commonly below probably even 15 or 25 feet. And they tend to like these rough water habitats with really high water flow, whereas Marginatus tends to prefer these calmer, deeper depths. And so some separation in terms of habitat there. I'm not gonna touch on this in the rest of the talk. I do have a study looking at this species across the whole range, but I'll just tell you the conclusion really quickly, which is it looks like we have a subspecies in the East Pacific that's morphologically different, so it has a redder coloration. So they are disjunct there, actually could be in the process of speciating. In the Red Sea, we have another species that's more closely related to the Pacific than the East Pacific one is, or the Indo-Pacific than the East Pacific. So Red Sea closer to the Indo-West Pacific. And then in Hawaii, this is actually at a population level differentiation. So although these individuals have made it here, there's some gene flow with part of the Pacific, but not across all the way to the Indian Ocean. So we see a lot of genetic structure of separation of the Hawaii population. And so this sort of makes you think about, in terms of how does a species like these guys both have really long pelagic larval durations. And so it's one thing to get to Hawaii, but it's another thing to stay, right? So those of you who are graduating soon are probably thinking about this as well, right? So if you came here and you want to stay, how do you stay? For these animals, you know, it's a matter of evolving that something about the pelagic larvae that allows you to stay in the currents and keep recruiting back to Hawaii instead of being just dispersed out into the open ocean. And that's right now is sort of an open question. We don't know that much about it. Okay, so as I was saying, initially, a lot of my focus was on management. So how can we manage these species? What can we use? How can we use population connectivity to inform species management? And so this is a graph. It sort of ends at 2004. It's really difficult to get data from DAR. I don't know if any of you have tried from the state. But so this is landings, main Hawaiian Island commercial landings of lobster species. So they grouped both spiny lobsters together. So this is all lobsters in the main Hawaiian Islands. And just note, you know, the top number here of pounds landed it's approximately 18,000 at the peak, right? And so you see this dip right here, and this is you also see this is divided by how the lobsters were caught, and this is actually a function of, if you just looked at this graph without catch, you'd say, oh, suddenly we weren't catching any lobsters. But really what happened here was there was a big regulation change, and all the lobster fishermen switched from using traps predominantly to using hand. And when you switch techniques, you know, it takes a while to figure out what you're doing. So that's this. They're ramping back up. So fishermen figure out what they're doing pretty quickly. And so in general, we've had sort of a steady catch through the main Hawaiian Islands. Now in the northwestern Hawaiian Islands, have you guys gone over that at all? The low-lying islands? Gotten through the geology. So I'll show you a pictorial of how much larger the northwestern Hawaiian Islands is than the main Hawaiian Islands. But in the northwesterns, you can see this is number landed in thousands, but it's approximately the same as pounds. If you think about a pound lobster is about the size of a legal size lobster. And so in the northwestern Hawaiian Islands, you know, this is almost an order, or it's more than an order of magnitude more that were taken out of there. And this is just of predominantly just this one species. And so this fishery opened up in the early 1980s and rapidly increased. This is actually, again, fishermen learning how to fish. And in this case, it was actually a lot of Alaskan crab fishermen who came down and thought Hawaii would be a great place to fish in the off season of Alaska because the weather's obviously a lot nicer, and they decimated this fishery. I mean, you can say what you want about fishing impacts on ecosystems, but fishing in this case combined with change in the environment and low recruitment over a number of years, and these stocks dropped rapidly within only 10 years. So this is one of the fastest declines in a fishery that we know of, an example. And so there's a long history in here, and I know Cindy knows a lot of it, but and in terms of establishing various levels of protection and closing the fishery down. But in the sum was that by 2006, that whole stretch of the Northwest was declared off limits. There were still a few bottom fish fishermen that were allowed to go up there until 2011, I think, but the lobster fishery was closed. And so this is about the time when I started my PhD, and I thought, hey, wouldn't it be neat to look at whether now we have this fishery closure, is there any chance that that's going to help repopulate stocks in the main Hawaiian Islands and help with the main Hawaiian Islands fishery? And so if you stretch the Hawaiian archipelago off across the continental U.S., you can see that the main Hawaiian Islands, they go about down from New Orleans over into, I think this is like Texarkana, great town, and then the Northwest stretches the rest all the way across Texas, Nevada, Arizona, into California, right? So it's a massive. It's hard to fathom when you're here, right? And you're just flying short between islands. But in terms of the distance between here and Kure, it's really far. So, like I said, the question was, does the monument, will this provide a refuge for lobster stocks throughout Hawaii? And if so, can we estimate sort of what rate of replenishment we could expect, right? And then do we see the same patterns between both species? And then based on whatever the results are, what management, additional or is this enough? We can say, hey, this is a great idea. Does it really help us out? Okay, so I've talked a lot about population structure, and we gotten to the point that we're going to use genetics, but how do we know that there's population structure? I think I just gave it away, right? So what you might hope is that those invertebrates could just tell us where they are, right? So for some fishes, we can tag them and look at and track their movement. But I don't know. There's only a few invertebrates that have gotten into Twitter so far. I don't know if you guys heard about the octopus that escaped the New Zealand aquarium. This is one hypothesis of where they might be. Octopus are known to like sporting events. We have that one that predicted all Germany's World Cup scores many years ago. So I think it's a valid hypothesis that this is where that guy went. But they're not actively tweeting. Not a lot of them anyway. And so we're resorting to techniques such as genetic markers. And essentially, I didn't want to go into the specific details of genetic markers, but essentially it's just using pieces of DNA that we can isolate from organisms. So for the invertebrates anyway, we're non-destructively sampling, taking a piece of tissue, extracting that DNA from that organism, and then you can look at familial history. So you're not necessarily getting at what happened in one generation, but you can get it over multiple generations depending on how diverse those genetic markers are, how many of them you have, and how good you are at tweaking the analysis. Okay. So to do this, we had to go out and collect lobsters, right? So we had to collect lobsters. So for the Panilaris marginatus species, these are the collections, so it's over 564 individuals, so just taking a small leg or a piece of the antenna from each of these to get tissue. And I sampled 13 islands or atolls, and this is across the whole range of the archipelago. And this was back before we had tons of DNA markers, which is the way things are now. And so I only used just a single marker, which in retrospect sometimes seems to be more informative. And then for the other species, for Penularis penicillatus, this is 281 individuals. Across here, we only got 10 islands or atolls, and it's still just a single genetic marker. And so, as I was saying, there's that span of the Northwest Hawaiian Islands is massive, and so this is sort of all of the people that helped collect along that, and all of the agencies, and that's just a brief. This is my quick acknowledgement slides, but to give you guys an idea, for those of you who are thinking of going into research or have to interact with researchers, this is the sort of efforts that are required to answer some of these questions about the world. Okay, so again, not going into any specific statistics, but for the first species, what we saw was there's a regional differentiation between the Northwest Hawaiian Islands and the main Hawaiian Islands. So again, we have this monument. This is the whole monument here, right, protecting lobster stocks. But we see that there's population structure between those two regions. In addition, I found that two of the islands, so sort of this is a Maui-Nui complex, so some of the samples are from Maui, but also from Molokai and a couple from Lanai, and then and Kauai are also distinct. So within those main Hawaiian Islands even, we have distinct populations of lobsters. In the other species, in Penularis penicillatus, we see the same break across the main and between the main and Northwest Hawaiian Islands, but I didn't see any other within island structure. And the last thing that we want to know, right, is how does this translate in terms of migration rates? So if there is structure, is there still, obviously there's some lobsters getting between, right? There's still a single species across the whole Indo-West Pacific. So there's some lobsters getting in different locations. So one of the ways we can look is to say, okay, what are our migration rates? So if we're separating population size from movement, can we get an idea of which way these organisms are going? And so if the migration is equal in both directions, that's a much different story than if the migration rate is much higher in this direction than it is in the other direction. Unfortunately for me, I wasn't able to resolve these patterns in terms of migration rates anyway, and that's one of the issues with using one marker. So by using multiple markers, you have a much better chance of getting at that. So then the other approach is to look at other species, and so to give you now more of a sort of broad overview of invertebrates in the Pacific, the lab that I was working on, we had a number of people working on both invertebrates and fishes to look at, can we get some idea of some generality across different species. And the sum is that, unfortunately, every species is different. So this is Derek Skilling's work, and this is Holothuria atra, so one of the sea cucumbers here, and he essentially found three distinct populations. So not both the lobster species aren't all at Johnston, but these sea cucumbers were. And so he found, again, that northwest to main Hawaiian Islands break, and then Johnston Atoll as a third population. So he's like, well, I'll look at another sea cucumber species and see if we get the same pattern. And here he found a single population across the whole Hawaiian archipelago, and then a connection with Johnston. So still a little bit of isolation with Johnston Atoll, but still better connected populations than in that first species. Another person who wanted to see, hey, can we look at multiple species and get sort of an idea, at least within a genus, of what's going on, was Chris Bird. And so for some of his work, he looked at the 3 Opihi species, so the 3 Solanas, and these aren't technically reef organisms, but they're in the intertidal, and they're located at 3 different depths in the intertidal zone. So Solana exorata is at the highest depth. So if you guys walk on the rocks, you might see these guys. The Solana sanochensis is in the mid-intertidal zone. So this is the Opihi alina. So generally in the pinker rock, these are the ones that are more considered more of a delicacy. And then Solana talcosa, I don't have a picture of, but they're the sort of the round ones, the kneecap Opihi that are located down in the subtidal. So that sort of habitat differentiation, maybe that can explain differences between species. And what Chris found is that he, again, found separate patterns for each species. And so we're still looking into this a little bit for Solana exorata, but it looks like there's actually each island, now we have some from Oahu, and each island looks distinct in the main Hawaiian Islands. There's another group, there's a separation between the Northwest and the main Hawaiian Islands for Solana sandwichensis. And then Solana talcosa was specifically isolated in Kauai. So Solana talcosa, the kneecap OPE, doesn't actually go any further than Kauai. So it didn't even make it any further. The range doesn't expand this far. And then Kauai is really distinct from the rest of the main Hawaiian Islands. So again, different patterns across each of those species. Anushka Fauci, who some of you may know through biology, she was looking at vermidid worms, and I'm not sure the final conclusions. I know she's still working on her project, but I do know that she's found different populations on every island. In some cases, she's found different species. It looks like they might be different species or subspecies, and she's even found different populations within, for instance, the Big Island, and kind of a funny story with two different populations on Oahu that looks like it was probably somebody who collected some on one side of the island and dropped a bunch of rocks off on the other side. But that's a longer story. Paul Simeon was an undergrad who came, and he was looking at Octopus cyanea, so taco, day taco, and he found specific breaks between the northernmost atolls in the Northwest Hawaiian Islands, and then a single population across the whole rest, and then another breakdown at the very southern tip between the Big Island and the rest of the archipelago. And I think this is my last invert example. Molly Timmers, who's now working on looking at whole communities off of settlement plates. She did a project on acanthaster, really wanting to look at outbreaks. So do you see acanthaster outbreaks in different locations throughout the Pacific, and are they driven locally, or are they driven by individuals coming from other locations? And for Hawaii, at least, she found that not only is acanthaster a single population throughout the whole archipelago, but also Johnston Atoll is included in that population as well. So, what do we learn about marine management? Does anyone have any ideas? Not a lot, right? And it gets worse. So once we add all the fishes on, so the inverts are they're the least studied of everything. The most species that have been looked at are the fishes. And so when you add the fishes on, you get every pattern under the sun, right? But this is the power in looking at multiple species. So when you sum up sort of these different patterns, then you come to the conclusion that, and so Rob is the main PI of this lab, and I'm stealing some of his slides, but when you come to the main conclusion that there's basically these 5 major barriers to dispersal in the Hawaiian archipelago. Right, and so this one is it's kind of a a broader barrier, but that's the one that's between the main and the Northwest. And so we see this pretty commonly, 14 out of 20 species. So does anyone think the monument is helping the main Hawaiian Islands that much? Was that like the best idea of where to put a big reserve? Still good, but a lot of species have different populations in the monument than in the main Hawaiian Islands. Right? So for some species, there could be gene flow and repopulation from the northwestern Hawaiian Islands, but for a lot of species, there's a break here. Here, up in the northernmost atoll, so this is Kure, Pearl and Hermes, and Midway Atoll, we see again 10 out of 18 species show a break there. The reason the total numbers are different is because not every species has a range across all these, or was collected across all these. Again, so 14 out of 20. 12 out of 21 show that pattern I was describing for the kneecap OP, right? So Kauai seems to be isolated from everything else. And then we have 8 out of 19 that show a little bit of a break between Oahu and the rest of the southern islands. And then again, this other big break, just like was seen for Taco, between the Big Island and the rest of the main Hawaiian Islands. And so these patterns range across species that, from dolphins, right, that actually are swimming as adults, but dolphins show distinct populations on the Big Island, all the way down to, those vermidid, there we go, the vermidid worms. And so in general, what we're finding is that a lot of recruitment is local. And this makes sense, right? Because if once these species got here, remember I talked about that in the beginning, they have to somehow evolve to stay here, or else they're going to be cast off into that vast extent of open ocean where they can't survive, right? And even though I wasn't able to find out from the lobsters, some of the other species were able to look at these migration rates using additional genetic markers. And we find that in general, that rate of larval movement is 3 to 30 times, depending on the species, higher going from the southeast to the northwest than it is going from the northwest to the southeast. So to kind of start to sum up those management implications, right? Now we can make some management decisions from this, right? We can draw these boundaries across different regions that seem to be separated somehow in terms of multiple individuals and their populations, or multiple species and their populations. And we see that most of the structure, the genetic structure, is between islands in the main Hawaiian Islands, right? So just like your terrestrial species, it seems like there's breaks between each of between the habitats of the main Hawaiian Islands. And this could be due to the channels. If anybody's done any of the channel crossings, you know how the currents rip across there, and they may cast out larvae, whereas here you have shallow reefs. It could be due to habitat, like the amount of habitat. We're not sure, and so those are kind of the next steps. I'm involved in a bunch of projects now trying to look at, can we look across all these species? So I think we're now up to 50 taxa that have been surveyed across this range, and we're trying to look, what are the characteristics? So does it have anything to do with reproduction or population size, or could it just be due to habitat, or is it due to ocean currents? And so that's sort of the new directions that we're going in. Okay, I think that's all I have. I'll take any questions if you guys have. Thanks.
So based on your these results out of the tune-in lab for the past however many years, has there been a shift in thinking about setting up marine reserves on each of the main Hawaiian islands to preserve these existing populations?
Yeah, I mean that's definitely a push. So I'm involved a lot with the OPE, especially, and the OPE show genetic structure on each island, and we're looking now, it might even be within islands, and so that's been a big push. Chris tried to put in legislation a couple years ago and got pushback from the ledge in terms of Oahu has no Solana talcosa anymore. Like it's really hard to find that species here, and so putting a ban on Oahu in general to try to let those populations come back, and a lot of movement towards letting communities manage their own fisheries, because it's the community that's out there fishing. They're the ones who know what's going on with the resources way more than we do, and so that's like there's been success right on Kauai, and now I've been working with Uncle Mac on Molokai to try to set this up. He wants to set up a reserve for lobsters over there in Mo'omomi, and there's efforts on the Big Island as well. So yeah, I think this is definitely going a long way to inform that type of matter. Cindy, yeah.
The samples that you extracted, is the DNA still around? Could you go back and look at more loci?
Yeah, it's great. It's only, so I have both of these species and a couple other species. We have now pooled libraries that are just waiting for funding to go on the sequencer. So I have some sequenced of just like the species in general to try to develop new markers, but it seems like it's more efficient now to just sequence with the next-gen sequencing. It just keeps getting cheaper and cheaper. For me, it's just a matter of getting those funds. But I was really impressed. I had DNA extractions that I had done in 2006, and they looked like they were done 2 days ago. So the DNA is preserved really well. Any students? No. Sure.
What's is it a Kona crab or something?
Yeah.
Is that a slippery lobster?
No, that's a reptanid crab. I think it's reptanid. Yeah, so it's actually I think
it's a deep water crustacean.
Yeah, they can be in shallow water, but generally in sandy habitats. Yeah. And no one studied the genetic connectivity of those yet, so if you want to take on that project. Cindy, sure.
Where are when you see the lobsters at Costco, where are those from?
I'm not sure. I think they're from, I've seen them from Brazil before, and I don't know if they bring them in from Florida, and I'm not sure where else. But yeah, those are the ones I've seen. So they don't get them from here. The lobsters from here, once that Northwest Hawaiian Islands fishery closed down, you see guys selling them on the roadside stands sometimes, but I think they're mostly used for home consumption. Yeah. Sure.
Maybe Maine?
The main ones, I haven't seen. Have you seen the main ones in Costco?
I'm not sure. I'm just guessing because that's kind of where everybody kind of thinks of, like where lobsters.
Yeah, I mean, so the thing about lobsters is if you're buying the whole lobster with the claws, those are from Maine for sure, or Canada, right? So, and they actually aquaculture those. They might have some of those in the Big Island as well. So in Kona, I think you're saying yes. Yeah, I think they started aquaculturing them there. And I don't know how big those have gotten, but if you see a tail for sale in any of the markets, they're definitely not the Maine lobster because they're just too valuable with the claws on. So all the ones that are tailed are the spiny lobsters, just because there is meat in the head of the spiny lobsters, but it's not enough that's worth the extra poundage and processing. So they just rip the tails and sell those. There is a guy, I don't know, for a while, I don't know if he's still around, he was on Craigslist was selling lobster tails, and they were coming from, well, one time I bought them and he told me Thailand, and the other time he told me Taiwan, and then I asked him, and I was like, well, where are they coming from? And he's like, well, it's the same. And I was like, no, it's definitely not. They were also tails that were about this big, like in terms of length, and maybe this wide. So they looked more like shrimp. So I could only imagine them catching like the smallest lobsters and bringing it. It had to be some sort of an illegal operation. I guess it just makes it better to not know what country they really came from.